Hey guys, my name is Max and welcome to Simple Biology. In this video we're going to be talking about genomes. Now the word genome means the sum total of all the genetic material within an organism's cells. Now a genome of course includes all of the genes that code for all the proteins that those cells and that organism will use. But there's also a lot more to the genome than just those genes. And so it's interesting that as you look at the genome as a whole, there's more than just those the parts that we've studied so far. And so in this video we're going to be talking about those other parts of the genomes and the genome as a whole and how that plays a role in an organism's life. So first we're going to look at some of just kind of the overarching differences between the genomes of different species. Now most of the differences are only really visible between eukaryotes and other organisms, but first off we have just the size of the genome. Eukaryotes have larger genomes than prokaryotes. A lot of this has to do with the length of genes and the complexity of different parts of the genes. A lot of eukaryotes have introns and exons, whereas prokaryotes don't have introns. And then there's just a lot more regulation of genes when we talk about eukaryotes. So as a result of all this, eukaryotes have larger genomes than prokaryotes. Next, eukaryotes have more genes than prokaryotes. This is due in part to the fact that eukaryotes are much more complex than prokaryotes, and they use a lot more proteins. And as you know, each gene codes for a protein, so if the eukaryotes are using more proteins than prokaryotes, they're going to have more genes. Now, of course, what's interesting is that eukaryotes actually have fewer genes than would be expected. As you start looking at the larger sizes of the genomes, the distances between the genes increases a lot more than the actual number of genes. So with eukaryotes, we say that they have a lower gene density than prokaryotes, which is kind of interesting that they're not utilizing all of that space. Also, eukaryotes have alternative gene splicing, which we talked about in our eukaryotic gene regulation video, and they also have further protein modification, both of which can play a role in the protein's overall function, rather than just the genetic material of the gene. So eukaryotes can get by with fewer genes playing multiple roles than prokaryotes. In prokaryotes, each gene is only going to code for one protein which has one function. Eukaryotes have a little bit more leeway where that is. Also, eukaryotes have much more non-coding DNA. I talked about this just a minute ago with when I said that eukaryotes have more regulation and stuff. Non-coding DNA includes introns, regulatory sequences, and a lot of other stuff, and that's what we're going to be talking about for much of the rest of this video. So, like I said, non-coding DNA. Non-coding DNA is just like it sounds, any DNA that doesn't specifically code for either a protein or some form of RNA that will later play a role as either RNA or as a protein. Yeah. What's really interesting is that only 1.5% of the genome actually codes for something specific. This is tiny compared to what we would think. As of yet, this is all that we've talked about, just that 1.5% of the genome. And so, as we see, there is a lot more of the genome that's available to be used for other purposes. And so, that's why we're going to be looking into those. So, next is there's actually 24% of the genome is made up of introns and other regulatory sequences. We've actually also talked about these. The introns are located in the middle of genes, and they're largely cut out and not used to translate into proteins. There's also many regulatory sequences that play roles in helping determine which genes are transcribed and which genes are not in the various cells of your body. And so those introns and those regulatory sequences make up 24% of the human genome. All these percentages are based on the human genome. We place a rather large emphasis on that because, of course, we are human. So I should have mentioned that earlier, but regardless, 15% of the genome is unique non-coding DNA, and a lot of this comes from previous genes that have been duplicated or mutated to the extent where they're no longer useful and they're no longer actually transcribed, but they still have those unique sequences in our DNA, and so 15% of our genome is actually made up of that, of various stuff that maybe once played a role in our evolutionary ancestors, but no longer is useful now, and it doesn't code for anything actually. Next, we have 44% of our genome, and this portion of our genome is repetitive DNA related to what's known as transposons. Now, transposons are sequences of DNA that can actually move within your genome. There's various enzymes involved in this. Some of them have like a cut and paste mechanism where they are taken out, the genome is kind of resealed, and then they move to another location and are inserted into that portion of the chromosome. Some of them have what's known as a copy and paste mechanism, and this actually utilizes reverse transcriptase, which is found in certain viruses, and actually suggests that transposons might actually have an origin from viruses that our evolutionary ancestors might have gotten, and those viruses have since become useless, and now they exist as transposons. Anyway, 44% of our genome is made up of these transposons, and this DNA is very repetitive because there is multiple copies of these transposons, sometimes hundreds of copies of just a single transposons. Lastly, 15% of our genome is repetitive DNA that's not related to transposons. These are just various really short snippets of just repeating sequence. Some of this exists in our telomeres, which play a role in preventing the decay of the ends of our chromosomes. Some of these are in the centromeres, which play a role in cell division. That's where kinetochores appear at the center, the kinetochores at the centromeres. That's what it's called. And there are just various other repetitive DNA sequences. A lot of these are actually used in DNA testing to just test like paternity tests or what have you. And this is just to tell different people apart. But these repetitive DNA sequences that are not transposons, they make up 15% of our genome. So next we're going to talk about genome evolution, and this is actually kind of an interesting idea of genomes evolving over time, genomes changing over time for a particular species, but that's what we're going to be talking about. So first off, the duplication of entire sets of chromosomes can provide a lot of new genetic material, or not new genetic material, but a lot of genetic material that can then be used in evolution for that organism. We've talked about this before. It's called polyploidy, but essentially when an entire set of chromosomes is added to that organism, it's actually more viable than other different mutations that can occur. And because of that, this is because of every single gene being present in the same proportion, but but doubled or tripled. And so this polyploidy, this duplication of entire sets of chromosomes, it can help the organism or the species have more genetic material to have natural selection occur on. Next, also over time, chromosomes can actually split or fuse, and this is over huge amounts of time. So not something that would just happen in a day, but for a species, two chromosomes might fuse end to end to form a new chromosome that's just a single chromosome, or a single chromosome might split into two more chromosomes. Every single species has its own characteristic number of chromosomes. For example, humans have 46 chromosomes, other species have their own characteristic number, but over time that number can change, and this is one of those things that can help distinguish when species have changed from one species to another, when a new species has been created. And so over time that chromosome number can change because these chromosomes can split or fuse. Next, when we have mutations and crossing over, this can actually duplicate whole genes and allow two copies of that gene to be present, so that one can mutate into a new, more usable form, maybe through natural selection and evolution. But those mutations and crossing over, that's what I'm going to show you right here. So these diagrams show you two ways that two chromosomes might line up. This is the correct way, where all the genes are lined up. You can see here the two blue lines represent two identical sequences. Often this will be a transposon that has transferred from one location to another, that's been copied from one location to another. And what might happen is that the chromosomes might line up improperly. So these are two identical sequences, but as you can see from the overall pattern, they're not identical sequences in the same place. So these two sequences have lined up incorrectly, and so if crossing over occurs in this little setup, then one of those chromosomes will have two copies of the red gene, and one of the chromosomes will have 0 copies of the red gene. And so by doing this, one of the offspring might have an extra copy of the red gene that can then be mutated into a new gene that may code for a protein that has some new function that's helpful for the organism. Last thing, exon duplication and shuffling, so of just the exons within genes, can create new genes. So essentially, if a gene has multiple exons with the introns inside of it, those exons can actually be duplicated, and they may be shuffled in a similar manner to gene shuffling. Those exons can be moved around into a new way that they can form a new gene that will code for a new protein. And the reason that this is interesting is because rather than it just being where genes are changing over time, those individual exons still code for something that's useful, still code for maybe just the transmembrane space of a protein, maybe just the active site of a protein, whatever it may be. But because those exons are being used in new arrangements, those new arrangements are more likely to have useful functions than starting over a new gene from scratch. And so that's really all we've got for genomes. Genomes are really just as simple as that.